This year, the annual Massachusetts Institute of Technology competition, iGEM, lured 6 South African students to break barriers in the field of synthetic biology.
The undergraduate students, or up to honours level in South Africa, have to get together as a team from different disciplines and develop a biological machine that performs a novel function. So we thought this was really interesting and really cool. So we got together and we kind of brainstormed a whole bunch of different ideas, and because most of the biologists on this particular team have a sort of medical bent, we wanted to address a medical problem that has a sort of South African flavour.
Human papilloma virus is the most common sexually transmitted infection in the world. Up to 85% of women contract this STD in their lifetime, which can progress to cervical cancer.
Most of the time your immune system will clear it and it won't progress on to cancer, but sometimes it doesn't, and then you end up over a long period of time after the initial infection getting cervical cancer. And what we found when we researched it is that it's actually quite a problem in the developing world. So people with a lack of access to healthcare, for example, that are unable to have regular pap smears, they have this problem where the disease progresses to cancer and it's undetected, and there are so many women, I think it's 250,000 women in sort of under-resourced countries are dying of the disease every single year. Well, what we thought is that the normal way of detecting cervical cancer is you have to go to the gynecologist and you have to have a pap smear. So for women in rural settings or poor settings, they're unable to do so. So we thought we could try and come up with this diagnostic that is simple, easy to use, affordable, and able to alert women that they've been infected with this virus that causes cervical cancer. And then if it's detected in time, they'll be able to seek therapy for it. And what we wanted to do is then take it even further and perhaps add a protective element to it, so they don't even have to seek care because our machine or our device would be able to protect them as well. So what we looked at doing is taking a bacteria that lives commensally in the vagina. So it's present in normal, healthy women all the time, and it lives there naturally. And if we could modify those bacteria to actually pick up on the presence of the virus and visibly change colour, so you would be able to tell immediately that you've been infected. And then developing the work to a later stage would be that the bacteria actually produce a protective molecule that blocks the infection. We were thinking of this population of bacteria in the vaginal mucosa. And the problem is that if one bacterium sees the virus in isolation and changes colour, you won't be able to detect that colour change. So for the competition, we decided to focus on a proof of principle study where we actually made the bacteria communicate with one another, so that if one bacteria on one end of the population detects the presence of the infection, it talks to all the other bacteria in the population and they all change colour at exactly the same time. So you get this massive colour change, and then you'd be able to actually visibly detect it.
Teammate Greg Mayer demonstrated the lab processes leading to the virus changing colour.
This is what a normal plate of bacteria looks like. And then once you've added the chemical, ideally it would be the human papillomavirus, but once you've added the chemical, it changes colour. And if you were to look at this under slow motion photography, you could actually drop in the chemical and watch this ripple effect spread, and you'd see how it changes colour.
The Witsies competed amongst the best universities in the world, including Harvard, Cambridge and Yale. As the first African team, organisers requested they encourage other Africans to participate in the future.
The whole idea is that it fosters the sort of spirit of collaboration and gets scientists from all over the world to actually get together and pool their ideas and try and innovate to solve problems.
The team hopes that next year's honours students will take the project to the next stages, to get the bacteria to detect the human papilloma virus and produce the protective molecule.